Welcome back to This Week in Weed. Let's take a look at our headlines.
Drug screening results, including those from federally certified labs, may not always be reliable, according to a white paper published online by National Work Rights Institute. Government-certified drug testing laboratories have significant reliability problems, and that the government's assurances that false positive test results are a thing of the past is untrue, the paper concludes. The NWI paper bases its conclusion on several key findings. These include: the accuracy of certified labs has never been tested. Government-certified labs do not consistently follow federally mandated procedures for lab accuracy. Federal regulations allow labs to make mistakes on 10% of the blind samples used in the certification process, and that certified labs do not always maintain a proper chain of custody. According to the paper, documented examples of errors committed by federally certified labs are not uncommon. It finds, in the last 4 years alone, one laboratory had its certification revoked and 3 others had their certification suspended. The activation of specific cannabinoid receptors moderates the progression of the human immunodeficiency virus, HIV, according to preclinical data published online by the Public Library of Science. Investigators at the Mount Sinai School of Medicine in New York City assessed whether the administration of a selective cannabinoid agonist could regulate HIV infectivity. Researchers reported that activation of the CB2 receptor inhibits HIV infection in culture. Authors concluded the clinical use of selective CB2 agonists in the treatment of AIDS may also exert beneficial adjunctive antiviral effects in the late stages of HIV-1 infection. The administration of THC is protective against neuronal injury in a human Parkinson's disease cell culture model, according to data to be published in the journal Neuropathology and Applied Neurobiology. Parkinson's disease is a degenerative brain disorder marked by involuntary movements. Investigators at the University of Plymouth in the United Kingdom assessed the protective properties of THC on brain cells exposed to PD-relevant toxins. Researchers reported that THC was protective against all 3 of the toxins tested in this study. By contrast, separate administration of the non-psychotropic cannabinoid, CBD, and synthetic cannabinoids were unable to elicit the same neuroprotection. That's it for This Week in Weed. Be sure to check back every Thursday for the latest stories in marijuana law reform.